Hi, my name is Matt Woodstock, and I'm a PhD candidate at Florida International University in Miami. Today I'm going to discuss an ecosystem model that we've developed for the oceanic Gulf of Mexico and use to assess potential shifts in the trophic structure of the system. So ecosystem modeling is an approach designed to incorporate all species that live and interact in an ecosystem, and the factors that influence energy flows throughout that system. The information necessary for the development of these models varies a bit by trophic level, where at lower trophic levels, we're more concerned with environmental impacts and hydrodynamic processes that influence nutrient availability. And at higher trophic levels, we're more concerned with life history characteristics and the influence of anthropogenic effects on the biota. So think of an ecosystem model as a collection of single species models that interact together within one simulation. To date, ecosystem modeling is more commonplace in coastal regions than in the oceanic realm. Well-developed ecosystem models can be implemented into management through the development of management strategy evaluations and marine protected areas. To date, this is less common in the oceanic realm, primarily because of data limitations offshore that we're all well aware of. However, popular ecosystem modeling objectives are of importance to our understanding of the oceanic zone as well. This means that the tools are developed to model oceanic zones. We just need the data to plug into the model. Considering the call to protect 30% of the world's oceans by 2030, ecosystem modeling may be a useful tool to make sure that the areas that are protected are the right areas from a conservation standpoint. Our focus is the Gulf of Mexico, and many of us probably remember the 2010 Deepwater Horizon oil spill that introduced an estimated 800 million liters of oil into the water column. So here's a map of the active oil and gas platforms in the Gulf of Mexico, as well as active leases to which new platforms could be developed. The Deepwater Horizon wellhead was approximately 1,500 meters deep, which was a major issue because at the time of the spill, there wasn't a method to cap a wellhead that burst that deep. Now, some of these leases are at locations seaward of the 3,000 meter isobab. The general conception is that the deeper you drill, the more dangerous it is. So the effects of oil exploitation in the Gulf of Mexico is very much a continuing event with the threat of more blowouts in the future. As for the hydrodynamics of the system, in 2019, Matt Johnson and others developed a classification scheme for water masses in the oceanic Gulf of Mexico. This animation is showing the daily classifications for the year 2017. There are two main influences in the eastern portion of the Gulf. One, the loop current that's shown in the dark blue, is an oligotrophic warm water current that enters from the south, occasionally develops mesoscale eddies, and exits in the east, becoming the Gulf Stream. We also have the Mississippi River, which dumps large amounts of nutrient-rich water into the Gulf of Mexico and occasionally increases the primary productivity that we see offshore. Our model domain is the northern Gulf of Mexico, bounded by the U.S. exclusive economic zone and the 1,000 meter isobab. Vertically, we're extending from the surface to 1,000 meters depth, encompassing the entirety of the epi and the mesopelagic zones. Since 2011, we've observed a massive decline in most mesopelagic fish and crustacean micronectin populations. Shown here is just the myctophid population for 2011 and 2015 through 2018, but other mesopelagic taxa show a similar trend. These taxa are major prey resources to commercially important organisms such as tunas, billfishes, and marine mammals, and they're also critical in energy transfer, as they're the dominant predators of the zooplankton community. A strange thing about this decline is the prey species for the micronectin have had a pretty static trend, so there has been prey for these organisms to eat. And there also hasn't been an increase in predator populations within the region, so predation pressure hasn't increased. Also, the environment has not significantly changed, with the exception of the 2010 Deepwater Horizon oil spill. Now, one thing that is different following the 2010 oil spill is the concentration of polyaromatic hydrocarbons, which is the signature of the oil spill in the environment. This was actually higher in 2011 than in 2010 in the muscle tissue of deep sea fishes. Between 2011 and 2015, that concentration in muscle tissues decreased near the pre-spill levels. However, there was still a high concentration in the guts and eggs, suggesting that the oil was now available within the food web. Given that many of these fishes are believed to live less than 5 years, we're talking about a multi-generational impact on the deep pelagic Gulf of Mexico. So this means that the oil spill was not a single pulse event that's in our past, it's an ongoing disturbance, and we want to explore that impact. So the question becomes, how does a decline in mesopelagic micronectin populations affect the rest of the Gulf of Mexico ecosystem? And we decided to focus on the potential shifts in the trophic structure for this particular project. To do that, the data we use are from sources such as survey data, fisheries-dependent data, life history information, and trait databases like FishBase. All of the mesopelagic data that's in this model come from surveys by the Deep End Consortium. And we used EcoPath with EcoSim, which is commonly used for these types of questions. An EcoPath model is essentially a snapshot of the ecosystem that incorporates all of the interactions among functional groups, including fisheries. It operates under the assumption of mass balance, where for all functional groups in the model, the amount of energy consumed must be allocated in some way, either through mortality, respiration, excretion, or assimilation. We can also incorporate confidence intervals surrounding input parameters in the event where we have little certainty in the life history of a functional group. Our model has 43 functional groups, including 3 marine mammals, 1 sea turtle, 1 seabird, 28 fishes, 7 invertebrates, 2 primary producers, and 1 fishery. And that EcoPath model allows us to characterize the initial trophic structure of the system and the importance of each functional group. This plot is the square root of the total system throughput, which is a metric that explains how important a group is to the transfer of energy throughout the system. Functional groups in our model are on the y-axis, and they're sorted by trophic level. So in general, you would expect low trophic level organisms to have a higher throughput value than apex predators that aren't consumed. And for our system, over 90% of that energy flow is the first 2 trophic levels, showing the importance of zooplankton and primary producers as prey in the oceanic zone. Given that most oceanic fishes, particularly mesopelagics, are unable to consume small-sized phytoplankton and particulate detritus particles in the ecosystem, zooplankton are also important predators of primary producers that essentially repackage nutrients in a vessel large enough for fishes to consume. In terms of where all this energy is coming from, our model suggests that 30% of the energy transferred through the ecosystem originates from detritus, which in our model is synonymous with marine snow. This indicates that marine snow is an integral part of the ecosystem, and it corresponds well to compound-specific stable isotope work by Christian Glocker around Hawaii and Travis Richards in the Gulf of Mexico that says detritus is an important component of non-migratory, lower mesopelagic and upper bathypelagic organisms. To examine how the system has shifted throughout time, we then simulated our model through 2018, forcing the population declines observed for mesopelagic micronectin. These models are deterministic, so a resampling procedure was used to incorporate the uncertainty in input parameters. And this allows us to track the succession of ecosystem processes over time. These are model results that again display all consumer groups organized by trophic level. The left plot is a trophic level, and you see that not much has changed between 2011 and 2018, indicating that the consumer groups were still feeding at the same trophic level. The trophic level of a functional group in an EcoPath model is calculated based on the trophic level of the preys. Now, however, biomass has shifted quite a bit, with most groups showing at least a slight decline. Although we don't see changes in overall trophic positioning, biomass shifts may influence individual trophic interactions, and that's what we want to investigate. So we adapted a network analysis technique developed by Ioanowitz and Piscia called the mixed trophic impact, which quantifies top-down and bottom-up impacts that each species and fishery have on each other, including the indirect interactions. With our 43 functional groups, we had a possible 1,849 interactions to assess. We took the model output from 1,000 iterations and reconstructed diet and mortality matrices at each time step, which for us was annually for 8 years. At each time step, we calculated the mixed trophic impact for each potential interaction to create a time series that was then assessed through a linear model. The code to do this is available on my GitHub, and I can provide that link to anybody that's interested. Throughout a simulation, each interaction could either strengthen, weaken, or have no observable change. These plots of the model output where small epipelagic fishes now provide greater bottom-up support to adult big-eye tuna. Hatchet fishes, which are mesopelagic fish where we've noted a decline, provide less bottom-up support for baleen whales, for example, the newly named Rice's whale. And there's no change in the top-down pressure juvenile bluefin tunas exert on lanternfishes, which I've already shown have declined. Overall, we saw over 1/4 of trophic interactions showed some change, with direct top-down interactions being the most labile. However, when one interaction strengthened, another one often weakened. We also observed a weakening of indirect trophic interactions, particularly those which provide bottom-up support towards higher trophic levels. This could be concerning, considering the commercial species in the oceanic Gulf of Mexico are upper trophic level organisms and rely on this bottom-up support from the intermediate trophic level groups. To investigate this further, we took a closer look into the parameters that are used to calculate the mixed trophic impact. This figure is the percent diet composition for adult big-eye tuna, with prey groups aggregated for simplicity. Big eyes spend a considerable amount of time in the mesopelagic zone and utilize mesopelagic organisms as prey. The proportion of diet composed of cephalopods and epipelagic fishes is now 16% higher than that at the beginning of the simulation, and mesopelagic fishes represent 10% less of the diet for this predator. Other upper trophic level species have a similar trend in diet, as mesopelagic micronectin population sizes have declined, indicating an ability for upper trophic level organisms to switch prey when one resource becomes less abundant. This corresponds to the idea that many of our oceanic top predators are opportunistic predators that are feeding upon a particular size range of organisms rather than on a particular taxa. And to show this further, we used a method from Anela Choi's 2016 ecopath with ecosim model of the Central Pacific Ocean, where we subset our diet matrix to only include those with a trophic level greater than 3.5, which was where we cut off intermediate and upper trophic level organisms. From our Bray-Curtis similarity matrix, we then identified the preys that most define the structure of the new data set. The colors in this plot are different predator taxa, and labeled vectors are the 6 most defining preys. In 2011, decapods, stomiids, and gonostomatids defined the majority of the trophic structure. Fast forward to 2018, and cephalopods and ichthyoplankton are still major preys, but the mesopelagic fishes are absent. In their place is 3 commercial fishes, which is likely the result of a depleted mesopelagic prey field. Moving forward, a reliance of commercial fishes as preys could negatively affect those populations, causing problems for management goals. Alternatively, and something that we haven't explored in this model, is the potential for top predators to just avoid areas that are no longer plentiful foraging grounds. There is evidence that sperm whales have avoided migrating to the area of the 2010 Deepwater Horizon oil spill footprint after the spill. Now, whether this is because of the prey field or other conditions is uncertain, but avoidance seems reasonable. Now, to give a perspective of micronectin trends, this is the relative predation mortality exerted on mesopelagic micronectin fishes by all predators throughout the simulation. As you can see, despite a decline in population size, predation mortality has not declined to the same degree, and was actually greater throughout much of the simulation. This indicates that the reduction in predation mortality required for a population to return to a previous state is not occurring within this system, inhibiting any sort of potential recovery. Euphausids have a similar trend to the fishids, but decapod crustaceans experienced a significant decline in predation mortality. This is likely due to the extreme population decline that they experienced, as well as decreases in the biomass experienced by other predators. A longer time series, which is in the works, will hopefully allow us to parse out the long-term effects of these declines. Now, before I summarize this talk, and in the spirit of this session, I wanted to give a preview into the other modeling efforts that we're working on. One is the development of a carbon export model for mesopelagic fishes, where we're matching our net caught assemblage to a model assemblage and applying species-specific traits to estimate bioenergetic rates and carbon transport across different flux boundary scenarios. This model also incorporates different feeding guilds and asynchronous vertical migration. We have a different ecosystem model in the works, created with the Osmos software that is linked to a biogeochemical model, where we're attempting to estimate the additional mortality that was exerted onto the micronectin community after the Deepwater Horizon oil spill. And finally, we're estimating the nitrogen that oceanic cetaceans transport vertically with the help of a stabilized stope mixing model. Now, each of these objectives have numerous offshoots that we would love to explore in the future. Investigations into the trophic structure of an ecosystem generally result in the development of a food web, but ecosystems and the populations within them are dynamic. So our ability to assess shifts in the trophic structure, whether caused by natural or artificial processes, is dependent on our ability to quantify interactions over time. In our model, that allowed us to see that a generalist feeding strategy by apex predators, which are thought to prey upon a diverse array of preys, provides stability when major preys are depleted. We were also able to see that the predation pressure on declined populations primarily scales down with population size, which could reduce the chances of recovery of a population. Now, the work I was just discussing is published, and I encourage you to check out that work and email me with any sort of questions or feedback. Now, finally, I'd like to thank all of those that have aided in the collection of the data that went into this model, and also those that have helped with model parameterization. Thank you.